There we go. So in terms of an overall objective, we want to produce novel varieties, and we're starting with native plant species. First priority is to start with native plant species from Canada, but we've expanded that over the course of the study. And we want to produce varieties that can stand the rigors of a variable climate, whatever that means to you. The tissue culture aspects of the project allow for rapid propagation of desirable genotypes once they are found. And then with the cryo preservation, it's a way to preserve genotypes, and there is some therapy that can go on that Dr. Praveen is going to talk about. And we also want to develop a system that will allow growers to easily work with tissue culture plants. Not everybody knows how to, has familiarity with this, okay? So specifically, we've started with native species to develop varieties in germplasm that are adapted to drought and low nutrient environments, looking at plants that are very resilient to climate change and can survive under low input environments. And we've heard during different talks today about water use efficiency, efficient use of nutrients, and this kind of builds on that. Praveen is looking at techniques to improve survival of tissue cultured plants, looking at things like optimizing the light quality during tissue culture, use of plant growth regulators, and a lot of emphasis on the culture vessels and bioreactors for improving in vitro propagation. And the final aspect is developing cryo preservation techniques. So this is storing plant parts in liquid nitrogen to not only preserve important genotypes. At GRIP, we work with a number of rare endangered species, but this also allows us to effectively store important ornamental species. And as I mentioned, there's a therapeutic function that cryopreservation has, and Praveen is going to talk about that. One of the other things that we do in this study is run trial gardens, and we run them at Guelph, Milton, and the Milton site is the Landscape Ontario site, and Royal Botanical Gardens. And I would say that we were quite successful running the gardens this year, despite the pandemic. There were, on our open houses, we had 300 visitors. This included people from the industry, landscape trades, and the public. This year, we also added a third open house. This was done in September to, and just was open to the major plant buyers for some of the major retailers so that they could see the new selections. Okay, so at these testing sites, we have the new releases for all of the major plant breeding companies that want to partner with us. And there's also the Ontario Perennial trial, which I'll elaborate on in a minute. Also at the Milton site, we run a pollinator plant research experiment with Sandra Genovisic. One of the things we measure there, and one of the questions we try to answer with Sarah's help, is, are some of these ornamental species attracted to pollinators? There's a lot of press that identifies native species, but what about the non-native species? And that's what this trial has is shown. So this is a picture of the trial garden at part of the trial garden at the University of Guelph. I just wanted to let you know that we moved to a new location in 2021. This is, it's still on campus on the Arboretum grounds, but associated with the new Guelph Turfgrass Institute. Roger Shantz, who provides technical support for the ornamental program, does a lot of work to produce and coordinate the trial garden, and he had to work very hard this year to get the site ready for planting. Okay, so at the Guelph site, we mainly have ornamentals and a few container trials. Okay, so at the top left, you'll see that's another picture of the Guelph site. The two top pictures are the Guelph site, just a range of ornamentals, and we, as I mentioned, try to get plants from all of the major breeding companies in North America. At Milton, we have a very large container trial, and that's addressing a very important need in the industry. Okay, and also at Milton, we have the Ontario Perennial trial. We have about 200 selections there, and this will be a long-term trial where we can keep the perennials growing in a habitat that allows their perennial nature to be measured. And of course, this is ongoing, and each year we're continuing to add to that. Okay. Roger coordinates all of the open houses. As I said, we've had people from the landscape trades, the greenhouse people, retailers, and the public, and it's been quite a successful venture, and it's an important type of outreach that we do. I think the added benefit that this gives us in the breeding program is that we can place our advanced selections in any one or more of these trials, solicit public opinion and opinion from the trades, and that can feed back into the breeding program. Okay, so let me talk specifically about the breeding efforts now. And as I mentioned, we're using Canadian native species to produce new varieties that are adapted to drought and low nutrient environments. On the left-hand side, you see ongoing studies. There's a number of species that we've been working with prior to the COHA grant, and we brought those in: Liatris, Thermopsis, Baptisia. And then there's new species that we've brought in, and this is just a sampling of those species that have been part of the breeding program. There's a little asterisk beside Lobelia cardinalis. That's a cardinal flower, and I'll talk a little bit more about that in a minute. But other Liatris species, Helenium, and Allium species. Okay. So this is the breeding protocol that we're using, and I won't go into a lot of detail here, but I want to point out that when we started the process, we planned to use sort of an S1 recurrent selection program. And the idea here was to self-pollinate varieties, choose the best plants, recombine them, and in a new population, and produce sort of a cyclic nature. As I mentioned, just repeat the cycle. What we found out in our first iteration of this scheme is that some of the species, and Lobelia cardinalis is one of them, is self-incompatible. And so it would not self-pollinate. That was a hard lesson to learn. But dealing with some of these native species, we don't know their genetic background very well, and we have through a bit of trial and error. So we went to a half SIB selection. So rather than selfing, we intercross all of the plants to produce a population and carry it through the scheme. One of the benefits of a recurrent selection program is that any point in the scheme, when you produce a population, you can then select elite individuals, and that's on the right-hand side, And you can start a slightly different breeding scheme where you're crossing the best with the best, testing these F1 families, and then selecting the top 1%. As I mentioned, these selections, and then it can then go into the trial garden system and then on to wider testing. Okay. So here's some pictures of the species from the long-term selection trial at the GTI. And this is a trial that's been in existence for upwards of 10 years. We've simply left it. There's been absolutely no input to it. And we go in and harvest seed from it. And you might say the plants that are remaining there are there because they're the most resilient, and they've been able to survive under the natural selection process, which again, I'll emphasize, is no inputs, no water, no fertilizer. Okay. And then we can turn these into populations that we can use for testing and selection. Okay. These are some of the plants that we've that have been part of our attention outside of this the previous trial. Penstemon, both of the Monarda species, both have attractive flowers, and you will recognize some of the common names as wild bergamot, spotted bee balm, and Amsonia, and eastern red columbine, Aquilegia, has both we're emphasizing in the program. One of the things that we want to do for during the selection process is be able to screen a large number of seedlings. And we're looking at two things. One is drought tolerance and salt tolerance. This is an example of screening for salt tolerance, where we just take flats of 50 plants, basically seeds from our populations, immerse them in these salt solutions on a biweekly basis, and select the plants that are the most vigorous. And and we can do something similar for drought screening, where we just simply withhold water from the plants. Okay. Here's a flat of 50 cell tray of Monarda fistulosa. The top tray represents seedlings that have been immersed in a salt solution. Okay. And we do this over several 2-week periods. And after about 4 emergence, we select the most vigorous plants, and you see those at the in the bottom slide. Okay. These plants are then moved to the field at Elora for further evaluation. We've done the same thing with Helenium. It would appear that Helenium is a little less tolerant of the salt, and you just see the amount of damage that's occurring. But there are differences, and we identified those, transplanted them to larger pots, and eventually took them to the field at Elora. And this is Thermopsis, same thing. I think the good news is that we were seeing differences. One of the things that I want to mention too is some of these plants are going to the field at Elora, but some of the plants also go into planting that we have with our partner, the City of Guelph, where we plant the plants on put the plants on a median, so division between 2 streets, that garners all kinds of salt, both from spray, the plowing during the season, and it's quite a harsh environment. Okay, the Elora Research Station is our main outdoor testing site, and again, we have no inputs here. Try to keep the field as clean as possible. We do some irrigation to establish the plants, and then and that's it. Okay. So we planted a number of plants in 2021, and it was difficult because the plants are so young. It was difficult to do a lot of meaningful selection, but there were some things that did stand out at the end of the season. This is Penstemon Digitalis, and I'll direct your attention to the plant in the middle of the picture. the one that's flowering. If you compare that to the plant that is higher up in the slide, the plant that we selected here appears to be resistant to mildew. So out of a whole row of plants, this one with mildewed plants on either side of it, this one was appeared to be resistant. So we were able to make some meaningful selections in this first year. This was Eastern Red Columbine, Aquilegia Canadensis. This has been a plant of interest to us for a long time, and we have a large number of seedlings in the field. This is one of those plants that to me appears rather delicate. It has thin stems, very ornate flowers, very attractive, but the reality is it's a very tough plant, and I think would do well in a number of settings. And so it continues to garner our attention. Right? Sort of newer to the program is Helenium. Has bright yellow flowers, very upright stature. I think one of its redeeming features is that it blooms very late in the season, so it's a good fall specimen. And we have a number of these plants at, we have a population of these plants at Alora. So going into 2022 season, what we hope to do is in the greenhouse right now, we are intercrossing plants that we selected from the field at Alora to make a new population. And these half-sib families, as we call them, will go into the field at Alora at some point, and I hope sooner in the season rather than later for future evaluation. At Alora, we also have seedlings of our long-term selections from the former GTI site, Baptisia, Liatris, and Thermopsis. Large populations of those are going into the field at Alora for selections. And from the plantings that we made in 2021, we'll make selections within those progeny for future testing and propagation. And just one of the things I'll add is that I was able to find a species called giant Trillium. It's Trillium chloropedalum. I think it's also called wake robin, great giant wake robin. It's a very attractive plant. I rescued it from a disturbed site, and we're hoping that we can get that into tissue culture and see what potential that has. So right now, I'm going to turn it over to Praveen to talk about the other three objectives. So I've stopped sharing my screen.
So I'm going to pick up where Dr. Sullivan left, and that was a really good introduction to what kind of species we are working with. In terms of the integrated program that Dr. Sullivan and I run at GRIP, the main component of research is Integrated Plant Production System, or IPPS as we call it. And it has three major components. The first one is plant germplasm. As Dr. Sullivan described, we are looking at native species and any other species that has commercial potential in Ontario. Once we have the germplasm identified, either from the breeding program or from natural collections, we want to produce as many copies as possible through micropropagation. Micropropagation is a technology that allows us to produce hundreds and thousands, if not millions of plants at will, provided that we have researched enough to have the technology in place. And then for germplasm conservation for the long periods, we use a technology called Cryo Preservation, which I will talk about in a couple of minutes. This technology essentially allows us to freeze the germplasm in liquid nitrogen at really ultra low temperatures of minus 196 degrees Celsius. And in that way, they are preserved for a very long period of time, almost indefinitely, actually. And they can be brought back to life and propagated again. We deploy these two technologies for both horticulturally important plants as well as endangered species. And then we utilize these plants for species restoration as well. At the same time, if the plant is of ornamental value, we would like to incorporate that in the overall scheme of things. So today I'm going to give you the report on the progress that we have made along these 3 lines in different plant species, and I'll give you examples of some of these species. Oh, I'm sorry. I think this. All right. This was the last one. So I'm going to use chrysanthemum as one of the first example because it does illustrate the micropropagation as well as cryopreservation. About a year and a half ago, it was suggested to us that chrysanthemum industry has a couple of limitations right now in Ontario. The first one is that most of the chrysanthemum varieties are virus infected. And the second is their production is not also very efficient for some of the varieties that are popular and should be not incorporated. In terms of micropropagation, this is how it is done. We can do it either in the liquid medium or in the solid medium. The photo here in caption B is a solid culture where tissues are nurtured on a medium that is solidified with the gel, and it contains the hormones and the micro and macronutrients. And here we have simply placed one bud and it has given multiple plants. And these buds can come from any of the popular varieties that the industry wants to propagate. However, we prefer to work with liquid medium for a number of reasons. The primary one being that liquid allows easy access of nutrients to the growing plants, and therefore the quality is improved. As you can actually see, this is a box with liquid medium. We put it in a rocker, which actually shakes the plant at the same time. And you can actually appreciate the high quality of these plants and the number as well. They both multiply in larger numbers, and it's easier to propagate and remove these plants from the boxes and transplant them in greenhouses. In order to achieve the high multiplication rate, we need to run several experiments for optimizing growth regulators, and particularly in relation to light as well. So here I'm going to present very brief data on one of the cytokinin which we find as most effective one is the benzylaminopurin. On the y-axis, you have multiplication rate and the shoot length, and then the BA concentration on the x-axis. And as you can see, the response in the liquid medium is higher in all cases, which basically proves that our hypothesis that liquid medium is going to improve shoot multiplication seems to be valid. We have developed a number of systems using different bioreactors to perform these experiments. But the summary of this is that we have more than 5 or 6 varieties of chrysanthemum in culture right now. And with minor variations, we can incorporate any other variety that may be of interest to the industry. This is the part 2 of the technology cryopreservation. We are using plants that we generate in liquid culture medium, and very briefly, we will put them in solid medium, although we can always use in liquid as well. It's just to give you a different condition of growth. I'm showing up a photo of a plant grown on solid medium, but we can always use the one from liquid as well. The shoot tips are excised and then they are plated. These are the nodal segments, and the reason is that we want to have a shoot tip isolated for cryopreservation process. Now you may wonder why go after shoot tip. Shoot tips are generally virus-free. They have much less load of viruses of infected plant. So if we receive a chrysanthemum germplasm that is infected with viruses, it will be possible to reduce the virus load if we excise the shoot tip. Now, shoot tip excision seems to be easy, but it's very complicated. It's kind of catch-22 situation. The shorter the shoot tip, the greater the possibility that it will have less viral load. However, the shorter the shoot tip, the more complex requirement it has for its growth. And sometimes we just fail to regenerate a plant out of a small shoot tip. So there's the balance which requires extensive research. Regardless, when we are successful, we can isolate the shoot tip and then we can dip it in liquid nitrogen. But from here to there are a lot of steps that we need to do. And I'm going to show you that in the next slide. But if the shoot tip is healthy, we will be able to dip them in liquid nitrogen, freeze them, and when we want to cryobank, we can place these into larger liquid nitrogen tanks. But for experimental purposes, we can dip them. They are as good as frozen forever. And after a few hours, we can take them out, or a few days or few weeks, depending upon the nature of the experiment. And then they are cultured again. So here is one set of observation we take, the number of shoot tips that survive, and then number of shoot tips that actually turn green and give us a complete plant. Once we have received this plant and we already have the micropropagation technology, then it's easier to multiply a virus-free plant. And this is the central theme of the integrated plant production system in case of infected plant species. So chrysanthemum is a really good example of this particular approach. So here is the protocol. I'm not going to go into too much detail, but from shoot tip to putting them in cryo tank, essentially the idea is to replace the water from the tissue for simple reason that if we do not do so, the water will form ice, the tissue will be damaged because the ice occupies more space, the cells will be ruptured. In order to do that, we treat the plant tissue on high levels of sucrose, for example, 0.5 molar sucrose in liquid medium for 1 day. And then over a period of time, we replace the solution with glycerol and sucrose. Again, these are cryoprotectant. Once they replace the water within the tissue on ice, they do two things. One, the explant is conditioned now to be frozen, and also the water content is much low. The explants are then ready to be immersed in liquid nitrogen. So here's some data to show cryotherapy. This is the therapeutic aspect Dr. Sullivan was mentioning, that with this process, we can eliminate the viruses. So here is the process that I mentioned and what it can produce. So as of today, we have around 40 to 50% regrowth after cryotherapy or the treatment with liquid nitrogen. And we are currently checking the levels of viruses. Unfortunately, we don't have too many infected varieties because there is a specific stage where they should be collected. So as soon as we receive them, we'll have more data to show. But anyway, the basic procedure works. And 40 to 50% is a very good recovery growth, because all that we need is 1 or 2 plants that have survived. We don't need 40 out of 100 population. Whereas think of this, we have hundreds and thousands of buds that we can freeze regularly. And out of that, 40% is a fairly significant number. In fact, even with 20, 30%, we are okay to proceed with production systems. Now, I said that it's very challenging to work with the meristems. So we thought not every plant is infected. We don't need to do meristem culture in each single case. So why not try to develop an alternate method where we can just take the plant tissue of different shapes and sizes. And here's an example of African violet, another very important plant. Here we isolated explant in 3 different sizes, and here's the protocol. These are the petiole explants, the leaf explants, and some of the microshoots as such. And we also had the somatic embryos as well, 4 different types of explant. They were frozen. And in all cases, we were able to get plants after cryopreservation. The efficiency varied a bit, but not very significantly within a group. So smaller explant, as expected, had only roughly 10% success. Plus LN is the liquid nitrogen treatment, minus LN is when the treatment wasn't given. So we can appreciate, just with the small explant, the 20 minutes exposure to cryopreservation can produce zero growth, but there's no liquid nitrogen treatment, then the regeneration efficiency is about 87%. However, still, the summary of this is even with small, medium, or large, anywhere from 20 to 40% efficiency is possible. This is one of the very novel observations. Most people tend to work with regenerative tissues in terms of meristems. Taking tissue, leaf tissue, or petiole has been done for the first time in African violets, and I think it's not very common in general either. This manuscript is in progress and will be published very soon. Now, this is the basic idea of integrated plant production systems that we are working with. And now I'll give you an example of how we are expanding these technologies to include other species that may have short-term and long-term interests, both from the industries and other government organizations. We are working with the 6 nations as well, and I'm sure you probably are familiar that working with indigenous groups is a very unique opportunity, but a very unique challenge as well, based on the traditional knowledge and the cultures and systems that needs to be or must be followed. We are very fortunate that we have been able to establish the connections with indigenous communities of the 6 nations. And we were given a couple of plans to work with to show that the technology works. And so we have done that, and we have given several seminars to the indigenous community, and they are now more than willing to work with us. In fact, our program is going to supply about 400 to 500 lobelia plants to the indigenous community for their assessment, and hopefully this will be an honor to work with them. So we will be bringing in more important species, and of course their medicinal plants as well, which also have ornamental values. Then there are other species that have recently been quite appreciated within the Ontario horticulture industry. Jasmine is one of them. Then there are different types of geraniums. It's one of the highly bred plant, and a number of selections are introduced every year. The process remains the same. I just wanted to show you that we are making a unique collections of very exotic ornamental species at GRIP. Also, we have extended the program to include the orchids, and the idea behind here is that there are many orchids that are endangered in Canada, and we have now access to them, like the lady slippers, and we should be able to incorporate them into the integrated plant production system as well. Some of the regular species also have quite a bit of potential. So I'm just going to take 2 or 3 more minutes, if that's okay, and conclude shortly. Another example of unique ornamental species, it's a spring flowering cactus, or the ornamental turmeric. Many of these plants are finding great popularity in Ontario horticulture market at this time. Each plant actually sells for $15 to $20. We have also extended the technology to the variants that we find in our trials. So here's an example of Baptisia. where we found a sport in the field, the blue one, and then it has been put in culture as well for further assessment. In addition to that, where the plants are producing seeds, for example, Dr. Sullivan mentioned about thermopsis, we have been able to mutagenize to alter morphology and appearance of this plant as well. And it has been cultured, and now we are transplanting that in the field for further assessment. So to summarize, this is the production system that we have. As I described before, micropropagation technology has been advanced to a number of species, and cryopreservation protocols are also in place, and we are expanding the collection from the breeding programs as well as the germplasm that we collect from nature. Thank you.